Good morning students. In this class you will study about reproduction in bacteria. In the last class you have studied classification of bacteria. It might be based on shape or based on nutrition. Before studying about the methods of reproduction in bacteria, so first you have to know some terms related to the cell structure in a bacterial cell. So you know that this is a diagram of a typical bacterial cell. A bacterial cell is made up of cell wall externally to the cell membrane or plasma membrane. So there is a presence of cell wall. Cell wall is made up of peptidoglycan. Okay, it is made up of peptidoglycan. But whereas you know that in plants, cell wall is also present, but that cell wall in plants is made up of cellulosic cell wall. External to the cell wall, there is a presence of capsule. Okay, so in some we could find there is a presence of the capsule, outermost covering layer. Within the plasma membrane, there is a cytoplasm in which there is a genetic material, bacterial genetic material called as nucleoid is present. What is nucleoid? Nucleoid is the genetic material, that is DNA, bacterial DNA, which is being coiled and directly found to be in the cytoplasm. Because there is no distinct nucleus due to the absence of nuclear membrane, the genetic material seems to be found in the cytoplasm. Okay, so that is called as nucleoid. And plasmid, apart from the genetic material, okay, so called as coiled genetic material, which we call it as a nucleoid. Apart from that, there is one more DNA present within the cytoplasm, extra chromosomal DNA. Okay, so we could find there is a small circular DNA present. A small circular DNA is present within a bacterial cell apart from the genetic material. So we can call it as extra. Okay, apart from genetic material. So that small circular DNA, we call it as plasmid. What is that important feature of a plasmid? It can replicate on its own, can produce one more copy without depending on the genetic material. Whenever it needs, okay, it requires, it can be undergo replication. Yes, that extra small circular DNA found to be in the cytoplasm is called as plasmid. Yes, small circular DNA. Yes, that is called as the plasmid. mesosome. What is mesosome? See here. In case of the bacterial cell, the plasma membrane or cell membrane, it is being enfolded like this. Okay, it is being coiled, enfold. Okay, that enfoldings of plasma membrane in a bacterial cell is called as mesosome. What is a mesosome? It is the enfoldings. Plasma membrane is pushed inside. It is being folded inside the cytoplasm. That enfoldings is called as the mesosome. What is the function of a mesosome? Okay, so mesosome, it helps in the respiration, exchange of gases, as well as helps in cell division. That you will come across in reproduction. Cell division itself is a mode of reproduction. So, mesosome function helps in respiration, means of exchange of gases, as well as helps in cell division. So, enfoldings of plasma membrane. Enfoldings of plasma membrane in a bacterial cell is called as mesosome. Pylori. So you can see here the hair-like structures all, yes, throughout the bacterial cell, these hair-like structures, so they are called as the pylori. Pylori are minute hair-like structures. What are pylori? They are hair-like structure. They are non-flagellar. They do not help in locomotion, movement. Okay, so we can call it as non-flagella. Flagella is present here. If flagella is present, it helps in movement. But these pylori do not help in movement. Okay, so these are hair-like structure. They are very minute, microscopic. So minute hair-like structure. Okay, characteristic feature, they are non-flagellar, they are proteinaceous in nature. Okay, what is it made up of? It is made up of protein called as pylene. Okay, it is made up of pylene. Hollow in nature, inside it is hollow. A water pipe you might have seen, only external it has. Okay, inside it is empty. A tube-like structure. Okay, a pipe or a tube-like structure, it is hollow in nature. So, pylori, it is a minute hair-like structure. It is non-flagellar, proteinaceous, and it is hollow in nature. Where is it found? It is present over the surface of a bacterial cell. Present over the surface of bacterial cell. What are the functions of a pylori? First function. It helps to transfer the genetic material from one bacterial cell to another bacterial cell. Transfer of the genetic material, DNA, okay, DNA from one bacterial cell to another bacterial cell. Transfer of DNA from one bacterial cell to another bacterial cell. Okay, as well as if a bacterial cell it is pathogenic, think this is a pathogenic bacteria, means what is meant by pathogenic? A bacteria which is involved in spreading, causing of the disease to the host organism. So then this bacteria is called as pathogen. Disease causing organism are called as pathogens. So if a bacterial cell is pathogenic, so then to attach to the host. Okay, so it will be used. Pili acts as a means of attachment to the host body, host tissue. Only if a bacterial cell is, okay, bacterial cell is what? Pathogenic. So, what is pili? Pili are hair-like structure. So these are some of the terms related to a bacterial cell. Okay, if the bacterial cells are motile, then they have the presence of flagella. They have the presence of flagella. Coming to the reproduction in bacteria, the bacteria produce offsprings, the young ones, by two different methods. One is by asexual method. Yes, and another is the sexual method of reproduction. But that sexual method of reproduction is called as parasexuality or parasexual method. Why is it called as parasexual? Later on you will study. Okay, so first we will study asexual reproduction methods in case of the bacteria. What is meant by asexual method of reproduction? So here there is no formation and fusion of gametes. In this method of reproduction, only one parent is being involved. So asexual reproduction involves binary fission and endospore formation. Asexual reproduction involves binary fission and endospore formation. There are two different ways of asexual reproduction. One is binary fission and endospore formation. How does this binary fission takes place and how endospore formation takes place? What is binary fission? Splitting up of mother cell into two daughter cell is called as binary fission. Splitting up of mother cell into two daughter cells. See here, only one cell. Okay, mother cell splits, only one individual is involved to produce the daughter cells, offspring. So hence binary fission is a mode of a method in asexual reproduction. Okay, splitting up of mother cell into two daughter cell is called as binary fission. Under favorable condition, a bacterial cell splits at every 20 to 30 minutes. At every 20 to 30 minutes, 20 to 30 minutes, a bacterial cell will split to produce two daughter cell. Okay, so this method of a reproduction is called as explosive growth. Okay, cell division itself is a mode of reproduction in case of unicellular. Okay, like bacteria. Bacteria, they are unicellular. Body is made up of only one cell. During time of binary fission, what takes place? It is nothing but the cell division. So hence we can call it as, it is called as explosive growth. Why? Because we could find in a short duration large number of individuals can be formed. This is why. Okay, at night time. Okay, so in a milk, if we put a drop of a curd, at the next morning we could find that the whole milk will be turned into curd. It is due to this. So at every 20 to 30 minutes, okay, at every 20 to 30 minutes, so we could find that a bacterial cell, a newly produced bacterial cell will undergo growth. It will increase in its size and undergo cell division and produce two new daughter cells. How does binary fission takes place? How does binary fission takes place in case of a bacterial cell? So you know that at every 20 to 30 minutes, okay, a one single bacterial cell, a mother cell, splits, divides to produce two new daughter cells. With a diagram, I would like to explain about reproduction in bacteria. What is this? It is a mesosome and nucleoid. A mother cell. So, steps involved in the process of binary fission. Replication of nucleoid, division of mesosome, membrane synthesis, wall synthesis. is a parental cell. Okay, so a parental cell, a mother cell, okay, undergoes the process of binary fission or cell division and produces two new daughter cells, and that binary fission takes place in 4 steps: replication of nucleoid, division of mesosome, membrane synthesis and wall synthesis. So now a mother cell, a mother bacterial cell, first step, okay, so first replication of, first step, replication of nucleoid. One genetic material, a nucleoid, will split, undergoes replication to produce its another copy. So now we can find in a mother cell there are 2 genetic material present. After this, so now second step, you can see here mesosome also splits and produces the another mesosome. So the second step is what? It is division of mesosome. Division of mesosome. Mesosome also divides and produces another mesosome. As it produces, it gets attached to the genetic material. Okay, so it gets attached during the to the genetic material. So I have told what is the function of mesosome. Mesosome is a site of respiration as well as helps in cell division. Cell division itself is a mode of reproduction. So this mesosome gets attached to the genetic material during the reproduction. So now what happens? After the mesosome gets attached to the genetic material, So now think this as a bacterial cell. So cell membrane starts moving inwards like this. So if you put a rubber band to this like this, what happens? It forms a constriction in the middle like this. Okay. So here after the genetic material which is being replicated, okay, that moves towards on either side of a cell, and by the help of mesosome which is being attached to the genetic material. So now the third step is membrane synthesis. The cell membrane starts moving inwards. When it touches each other, it will divide, okay, a mother cell into two daughter cells. Okay, so third step is membrane synthesis. The third step is what? Membrane synthesis. After this, yes, the fourth step we could find so now we could find due to the, yes, after the membrane synthesis, there is also the cell wall also splits, and we could find separate two new daughter cells. The fourth is wall synthesis. Wall synthesis means cell wall. Okay, so now you could find here a parental cell produces two new daughter cells. Okay, there will be production of two daughter cells from a single parental cell. Okay, so at every 20 minutes, okay, at every 20 minutes, one bacterial cell produces two new daughter cells. And there are four steps involved: replication of nucleoid, division of mesosome, membrane synthesis, and wall synthesis. And the second method in asexual reproduction is endospore formation. Endospore formation. What is endospore formation? Endospore are the seed-like structures. Spore means seed-like structure. Why is it these spores called as endospores? Endospores, these spores are formed inside. Endo means inside. Okay, so hence it is called as endospores. When are these endospores formed? These are spores are the seed-like structure, seed-like bodies. During the adverse condition, what is meant by adverse condition? Extreme conditions, unfavorable conditions, harsh conditions like scarcity of water, high temperature, okay, or lack of food. So these are considered to be unfavorable conditions, adverse condition. During adverse conditions, we could find the spores are being formed. How do the spores produced? So endospores are the resting spores, thick-walled resting spores. They are thick-walled resting spores. Thick-walled resting spores. On the occurrence of unfavorable conditions in the environment, see here, this is a bacterial cell, a rod-shaped bacterial cell. So what it does? There is genetic material. So around the genetic material nucleoid, it starts accumulating, collecting the protoplasm and starts producing out it starts producing out the membrane. A thick wall is being produced. On the occurrence of unfavorable condition, see here I am explaining how is endospore formed. When the conditions becomes extremes, adverse condition, that time the living material, the living substance within a bacterial cell, that is genetic material and protoplasm, starts accumulating around the genetic material, and around that there is one thick membrane, okay, wall is being formed, okay, and this starts to form as the spore. Okay, so the spore is formed inside a bacterial cell, so hence it is called as endospore. So we call it as thick-walled resting spore. Why is it called as resting? To overcome unfavorable condition. Okay, so the bacterial cell will be present in the spore condition itself. When the conditions becomes favorable, that time only we could find a new bacterial cell will be produced from this. Okay. So now what happened? The other part starts disintegrating. So it gets disintegrated like this. It will be removed. So now a thick-walled resting spore is being produced. And endospores are means of perennation. So what is perennation? Means of survival during unfavorable condition. See, when condition becomes unfavorable, the organisms cannot die. Okay, it has to survive. How it can survive? It will choose some methods, some ways by which it can overcome unfavorable condition. So this bacterial cell, to overcome extreme condition, adverse condition, it produces the thick-walled spore. This thick-walled spore is highly resistant to high temperature. Okay, it can withstand that high temperature. It can withstand the chemical supply, or we can find unfavorable conditions to the high temperature, chemical, or if we supply radiations also, it can withstand, and it is very light in weight, okay, due to the low water content. As well as if comprises of a chemical called as DPA. What is DPA? Dipycolinic acid. Not only due to the low water content, as well as a chemical called as dipycolinic acid. It is resistant to high temperature radiations, chemicals, and it is very light. It can be carried by the air. Yes. Now when it is carried by the air, falls on a suitable substratum base. Okay, when the conditions are favorable for the bacterial cell, that time what happens? From the environment, surrounding environment, this spore, endospore, what it does? It absorbs moisture, water. Okay, as it absorbs water, the spore wall breaks open and there will be substance, a living substance present, that is genetic material is there and protoplasm is there. That comes out. Okay, that comes outside and forms as a new bacterial cell. A new bacterial cell, from one bacterial cell only, a new bacterial cell is being produced. So hence endospore formation is also considered as a method of asexual reproduction. Though this is formed during unfavorable condition, adverse condition, so but it is considered as a means of asexual reproduction. Once again, I will tell, endospores are the seed-like bodies. Spores are seed-like bodies, seed-like structures. Okay, endospores are the thick-walled resting spores. Why we call it as resting? Until the conditions becomes favorable, a bacterial cell will be present in this state only, this condition only. Okay, it is very light in weight. Yes, low water content, very light in weight, and due to the presence of dipycolinic acid, it can, yes, so it can withstand extreme conditions like high temperature and all. It can be carried by the air when falls on a suitable place, and it produces a new bacterial cell, and it is mode of perination. Perination, means of survival during unfavorable condition. Okay, so this is called as endospore. Okay, how is it formed? You know, and it is formed, this spore is produced inside a bacterial cell, hence it is called as endospore. So here in this class you have studied the two methods of asexual reproduction. In the next class you will study about the sexual reproduction in bacteria. Thank you.